In 1999, Don Merton set out to find Kakapo that were presumed dead, but instead of evidence of their extinction, he found what became the beginning of a large-scale operation to save the species. Today, there are 8 such races. They are either returning from the edge of extinction or already trying to return through genetics, science, and people who simply refuse to give up. So who comes back first? On the Chatham Archipelago, tiny islands 800 kilometers from New Zealand, lives a bird found nowhere else in the world. The black robin, a small sparrow-like bird with black feathers and orange legs, endemic to these islands since the beginning of time. By 1980, 5 remained, and only 1 breeding pair, a female named Old Blue and a male named Old Yellow. They were around 9 years old, nearly twice the average lifespan of the species. The math was simple and brutal. If nothing changed, in a few years there would be no birds at all. And then Don Merton appeared, the same man from the opening of this video. He already knew what an absolute edge looked like. He had watched species disappear daily before his eyes, despite every effort. Merton took Old Blue's first clutch of eggs and placed them in a tomtit nest, a closely related species nesting nearby. The tomtits incubated the eggs and raised the black robin chicks. Old Blue, freed from her first clutch, laid a second, 2 clutches in a season instead of 1, and the math changed forever. She died in 1983 to 1984 at around 13 to 14 years old, a record for the species. From 5 birds, the species gradually began to grow, first dozens, then hundreds. Today, there are around 350 black robins, and every single one of them is a direct descendant of 1 female. The entire population of a species, from 1 bird and 1 decision, but 350 is at once a triumph in a sentence. All of these birds carry nearly identical DNA. Among them, there is no genetic diversity that would allow the species to resist disease. 1 virus passing through the population, and Merton's math could reset as quickly as it began to climb. The species exists, but it still holds on at the edge. In April 2025, the scientific community paused for a few weeks. A company announced that it had brought back from the dead a predator that had not existed on Earth for 12,500 years. Half of scientists said yes. The other half said, this is not what you think it is. Romulus and Remus were born October 1st, 2024. Khaleesi on January 30th, 2025. 3 living animals, white, massive, with a characteristically heavy jaw, and the company announced this as the first DE extinction in human history. The animal in question was the dire wolf, Enoceon dirus, a predator that went extinct approximately 12,500 years ago. But immediately after the announcement, scientists unaffiliated with Colossal began speaking out. Dusko Ilic, a professor of stem cell science at King's College London, called these animals synthetic proxies. His argument is simple. Not a single molecule of ancient Enoceon dirus DNA physically entered the genome of these animals. Colossal took DNA from a 13,000-year-old tooth and a 72,000-year-old skull, mapped the differences between the dire wolf and the gray wolf, and rewrote 14 specific gray wolf genes according to that map. Paul Wilson, a wildlife geneticist from Trent University in Ontario, described the result this way. In my opinion, they are currently creating interesting zoological novelties. The technology behind this is real and important, but the question remains open. If you take a gray wolf and change 14 genes in it, is that the return of a species or something qualitatively different? There is no consensus among scientists yet, and this video won't provide one either, because an honest answer doesn't exist yet. Romulus, Remus, and Khaleesi are already alive, but what exactly they represent is an open question. Colossal rewrote genes in a laboratory and produced 3 living animals, but there is a different kind of return, where nothing needs to be edited because the animal simply never disappeared. It was just hiding. And in 2023, it was finally found. Jordan Boersma, a researcher at the Cornell Lab of Ornithology, and his team spent a month on the island of Ferguson in Papua New Guinea, a place so remote that most people have never heard of it. The goal was the black-naped pheasant pigeon, last seen by scientists in 1882, once in the 19th century. After that, silence for 140 years. The team set up 20 camera traps on the slopes of Mount Kilkerran and another 8 at spots where hunters had once heard an unfamiliar call. It was a hunter named Augustin Gregorio from the village of Duda Ununa who suggested where to look. He remembered a sound that no one else could name. A few hours before the flight home, local team member Doc Nason reviewed the footage from one of the cameras. "When I saw the photo, I was stunned," he said later. In the image, a bird with a black body, a broad tail, and cherry red eyes. John Mittermeier, co-leader of the expedition, described the moment this way. "After a month of searching, seeing those first photos felt like finding a unicorn." The bird is alive, but what comes next is an open question. The population, according to the researchers' estimates, is very small, and logging companies have already begun to enter Ferguson Island. Rewild and local organizations are working to secure official protection for the area, but so far without result. The find resolved one question and immediately raised another. Will people manage to protect the habitat before it disappears along with the bird? In the autumn of 1981, a dog named Shep brought the owners of the Lazy BV Ranch in Wyoming a dead animal. The family didn't recognize it and took it to a taxidermist. He looked at it and immediately called the local wildlife service. It was a black-footed ferret, a species considered completely extinct since 1979, when the last known individual in captivity died. 2 years had passed since then, and suddenly a dog brings one in from the field. Biologists arrived at the ranch and found a living population of more than 100 animals. A species that had been written off was alive, but the joy didn't last. The ferret feeds almost exclusively on prairie dogs, rodents that once covered the plains in millions of burrows. By the time of the discovery, the prairie dog population was already declining due to plague, a disease introduced from Europe in the 19th century. Without them, the ferret cannot survive in the wild. Scientists launched a captive breeding program. For decades, they raised ferrets at centers across North America and waited for the prairie dog situation to improve enough to return them to the grasslands. In 2016, the first animals were released into the wild, symbolically at Matitsi, the very place where Shep had found the first one. But in 1988, before the breeding program had gained momentum, a veterinarian named Oliver Ryder asked to take a small tissue sample from a female ferret named Willa, just for storage. Willa never reproduced in captivity and soon died, but 32 years later, those cells were taken out of a freezer. In December 2020, in Fort Collins, Colorado, Elizabeth Ann was born, a clone of Willa, the first cloned animal from the U.S. endangered species list. Willa's genome contains 3 times more unique genetic variations than any living ferret today, because all living animals descend from just 7 founders. Elizabeth Ann cannot yet produce offspring, but her existence means that Willa's genetics did not vanish with her. At the Hobart Zoo in Tasmania, on the morning of September 7, 1936, a dead animal was found. The cause: staff had forgotten to close the enclosure against the night frost. The Tasmanian Parliament had declared the thylacine a protected species 59 days earlier. The zoo didn't even notify the press of the death. They expected to simply find a replacement from the wild. The name Benjamin, by which this animal is called in most sources, is a fiction. In 2022, researchers confirmed that the last individual was female and that no Benjamin had ever existed at the zoo. The name was invented by a former keeper in a newspaper article in 1968, 32 years after the animal's death. The Tasmanian tiger was a marsupial predator combining the features of a wolf, a tiger, and a kangaroo. Today, Colossal is trying to bring it back and says the project is ahead of schedule. The key partner in this is the fat-tailed dunnart, a marsupial weighing around 40 grams. At first glance, an absurd choice. How is this tiny creature supposed to help restore a dog-sized predator? The answer lies in marsupial biology. Their young are born extraordinarily undeveloped and spend most of their development outside the womb, in the pouch. This means a surrogate mother doesn't need to carry a fully formed animal. She carries an embryo. A separate team of 17 people is developing an artificial womb that has already grown a marsupial embryo to more than half the gestation period. But the transition from embryo to a living joey in the pouch, the critical step, has not yet been achieved. The progress is real, but slow. In 2022, Colossal, together with the TIGRR lab at the University of Melbourne, led by Professor Andrew Pask, began with the most basic task: reconstructing the thylacine genome from ancient samples. By October 2024, they announced that they had assembled a genome of approximately 3 billion base pairs with over 99.9% accuracy, the most complete ancient genome of any species in history. They even extracted RNA from a 110-year-old thylacine head specimen preserved in alcohol. RNA degrades far faster than DNA, and the fact that it survived is itself a scientific surprise. In parallel, the team learned to control ovulation in the fat-tailed dunnart, for the first time ever achieved in any marsupial. This is critical. Without predictable ovulation, it's impossible to retrieve eggs for subsequent editing. They then grew fertilized dunnart embryos in an artificial womb to half the gestation period, further than anyone had taken marsupial embryos before. But the transition from a dunnart embryo to a living thylacine is still several unsolved steps away. Independent scientists are waiting for complete data before drawing conclusions. The thylacine remains in the laboratory, but the laboratory has advanced further than at any point in the 90 years since that night in Hobart. The thylacine disappeared because of an unlocked latch, but there is a species that disappeared more slowly and is returning just as gradually, and there is a person who has devoted 50 years to that return. But first, the most ambitious project on this list. Colossal Bioscience's goal is a living woolly mammoth calf by the end of 2028. As of early 2026, the company reported that several cell lines have already undergone most of the planned gene editing. For more than 2 years, researchers have been working on combining the reproductive cells of the Asian elephant with edited material. The Asian elephant is the mammoth's closest living relative, but is itself endangered. Every surrogate female is a risk both to the program and to the species. And then there's the math. An elephant's gestation lasts 22 months. If the first attempt fails, that only becomes clear after the birth or death of the calf. After that, there is new preparation, new fertilization, and another 22 months. One failed cycle is nearly 2 years back to the starting point. In parallel, the same team of 17 is working on the artificial womb. In May 2026, Colossal announced that they had cleared all major technical hurdles except one. The system works like a dialysis machine. Algorithms read chemical signals from the embryo in real time and robotically deliver the necessary gases and nutrients. CE. Ben Lam put it this way: "We don't want to do 1,000 IVF procedures with elephants, but ultimately we want to grow thousands of mammoths." The first challenge is the Asian elephant herself. The Asian elephant has never been fertilized through IVF, not once in history. This means Colossal is not just bringing back the mammoth, but is simultaneously solving a problem that reproductive medicine has been unable to crack for decades. To implant an embryo, a female is needed who is physically and hormonally ready, and the species is endangered. Every female counts. The second challenge is the artificial womb. Colossal is building it in parallel specifically to remove dependence on living elephants. Technically, it has already grown embryos of small mammals, but a mammoth calf at birth weighs around 113 kilograms. By comparison, current artificial wombs can support premature lambs for a few weeks. The scale differs by orders of magnitude. The third challenge is the genes themselves. In March 2025, Colossal showed woolly mice, rodents with coats edited to reflect mammoth characteristics. The goal is to prove that cold resistance genes work before transferring them into an elephant. David Gold, a professor of paleobiology at UC Davis, said plainly, "Producing several mutations simultaneously in large animals is an entirely different level of complexity than in mice. Every gene that performs well in a mouse still has to prove itself in elephant cells. To hit the 2028 target, an embryo needs to be implanted around early 2027. That means right now, as this script is being read aloud, Colossal has less than a year to solve at least one of these three problems. And while Colossal battles with the mammoth, the same company is simultaneously working on a bird that became a symbol of extinction before humanity even knew how to say the word." The dodo disappeared in the 1680s, 70 years after Portuguese sailors first set foot on Mauritius. It did not fear people. Sailors picked it up off the ground with their hands. Bringing it back is harder than bringing back the mammoth. In 2022, scientists sequenced the dodo's genome from a museum specimen in Denmark and discovered that its closest living relative is the Nicobar pigeon. But cloning a bird is impossible in the same way as a mammal because bird eggs are opaque, and the embryo begins developing before the egg is even laid. The only path is through primordial germ cells, which are edited in the laboratory and introduced into a surrogate bird. The problem is that bird cells die outside the body within hours. Colossal tested more than 300 combinations, and in September 2025 found conditions under which pigeon cells survive and divide. In Texas, a colony of Nicobar pigeons is already living, alongside a flock of edited surrogate chickens. The plan is to introduce the cells into the chicks, raise them, breed them, and from their eggs, something resembling a dodo should hatch. Whether that will be a dodo is a question just as open as the one about the dire wolf. Kakapo once lived across all of New Zealand. There were so many that Maori hunted them without restriction. A large, fat, flightless bird that doesn't even flee from danger but freezes in place. The perfect prey. Then came European settlers with cats, rats, and stoats. Within a few decades, Kakapo vanished from most islands. By the mid-20th century, scientists were only recording them in the most remote fjords of the south. Don Merton spent years there. He found tracks. He found feathers. Sometimes the birds themselves. But every time, only males. No females. No nests. The species was considered functionally extinct. In 1977, on Stewart Island, his team came across a new group, an estimated 100 to 200 birds. Merton felt something close to triumph. 100 birds, that's a chance. Again, only males. Then the team brought sniffer dogs to the island for the first time. In 1980, a spaniel picked up an unfamiliar scent and led its handler to a smaller, slimmer, greener animal. Merton examined it and understood. The first female kakapo documented in more than 80 years. Soon they found 4 more, and nearby, nests and chicks. The joy didn't last. Feral cats on the island began killing birds one by one. Merton watched the population he had just found shrink again. Over the following decade, every known kakapo was captured and evacuated to 3 predator-free island sanctuaries. It was the only option. In the early 1990s, a full count was conducted. 51 birds remained. 51. Merton put the program on track and immediately ran into the first problem he hadn't anticipated. Kakapo breed only when rimu trees fruit. Not every year. Once every 2 to 4 years, when nature decides. The chemical composition of the rimu fruit acts as a hormonal signal. Without it, females simply don't enter breeding condition. The program could come to a standstill for years, and nothing could be done. But even in seasons when the rimu did fruit, 40% of eggs turned out to be infertile, and another 20% contained embryos that didn't survive. The cause was inbreeding. All 51 birds were genetically close, and each generation worsened the situation. The team began tracking the genome of every bird to match pairs with the greatest possible genetic distance. When natural mating produced the wrong pairing, scientists resorted to artificial insemination. Drones transported sperm between islands in minutes, a journey that had previously taken hours on foot. In 2019, aspergillosis broke out on Codfish Island, a fungal infection that killed 7 kakapo. The program didn't stop. Every bird wears a radio transmitter, every egg is monitored, eggs are frequently removed for artificial incubation, and chicks are sometimes raised by hand. When a female leaves her nest at night to feed, the system registers it automatically, and a ranger descends to check the nest. Don Merton retired in 2005, but the program he launched kept growing alongside the birds. Merton died in April 2011. At that point, the kakapo population had reached 131 birds. He saw the species rise from 51 to over 100, but did not live to see what happened next. In February 2026, the rimu trees decided to fruit. Females settled onto their nests. On Pukenui Island, the first chick of the season hatched, then another, then another. On Wenuahau, veterinarian Adam Naylor found a chick showing no signs of breathing and gave it mouth-to-mouth, gently breathing into its beak until it drew its first breath. By April, there were 106 such chicks, a record in 30 years of the program. Merton died in 2011 when the population had reached 131 birds. He did not live to see 106 chicks in a single season. The program continues.